All right, so now on to PCR. And PCR is the basis for much of this technology. So this is really going to be super important for many of the other things we're going to talk about as well. Polymerase chain reaction. It was a technique devised in 1983 to amplify small amounts of DNA by Carey Mullis. It can be performed on a single cell's worth of DNA now. And the amplified DNA can be used to do a bunch of things. You could use it to identify a suspect in a crime scene, determine sex, determine species, determine paternity, or you could use it in other biotechnology processes to start things like gene cloning and gene therapy. So it's a very, very important part of this process. So what you need? First, you need DNA. So if we're talking about a crime scene, you need a sample of DNA from the crime. Now, that might sound easy. However, one thing I want you to remember, we went over this like a long, long time ago. Red blood cells do not contain a nucleus. And so because they don't have a nucleus, they're not going to have DNA. So most of the time you have plenty of white blood cells in a blood sample that you can get DNA from those. But that's just one thing to kind of consider. The enzyme you're going to need is called Taq polymerase. This enzyme works at high temperatures because it comes from an organism that lives in hot springs. And so we take advantage of this high temperature thing in the process of doing PCR. We're basically taking advantage of how Thermus aquaticus, this organism that lives in these hot springs, does the normal processes of DNA replication. So we're basically doing DNA replication using this specific polymerase. We also need a primer. A primer is going to attach to the place we want to start the copy on the DNA. And then nucleotides, because we're going to need to add those to make the copy. If we have our DNA we want to copy, so we'll just say it's this ATTAGCGT. That's our DNA we want to copy. We have to have enough T's and A's and C's and G's floating around so that they can go bond with this, right? And then we need some way of making the temperature changes. There's a couple other things you want to have in your buffer. You want to make sure that it's like a neutral pH. You want to make sure there's liquid in there for all this stuff to float around, but that stuff is not usually as critical as these other components. Now, initially, when this process was first started, you would have your tube at different temperatures, and you have them there for maybe 30 or 45 seconds, maybe a minute or 2 minutes if it's a long strand of DNA you want to copy. But the original way we did the temperature changes was literally you had 3 different like water baths on different hot plates. So here's our 3 hot plates. You've got your thermometer in there, making sure that each hot bath stays the right temperature, right? And then what you would do is you would have like a grad student or an undergrad student stand there and put the tube in this one and have a timer for 30 seconds, and then put the tube in this one and have a timer for 45 seconds, and then put the tube in this one. And they would have to do that for 30 rounds. So after it was in this one, then you'd go back to this, then you'd go back to this one and start all over again. So this was a very like labor intensive process. Now we have machines that you put the tube in the machine, you turn on the machine, and it will rapidly go up to temperature, then down, then up to whatever temperatures it needs to go to. And we'll kind of talk about that. We're going to do a lab where you'll see this, a lab on the computer where you'll see this. We were going to do a lab in the lab, but unfortunately that won't happen this year. All right, so here's the basic process. So step 1, we have to denature the DNA. So DNA will start to just basically fall apart at high temp. So we heat it up to 95 degrees C. This is very hot. Remember, 100 degrees C is boiling. So this is very, very hot. Step 2 is annealing. This is where our primer is going to have to attach. So usually we'll have 2 types of primers. One that's going to attach here to go this direction, one that will attach here, because remember our copy is going to be going 5' to 3', right? So step 2, annealing. We have to lower the temperature back down to about 55 degrees C in order to get those primers to just naturally pop on. Then step 3, synthesizing. We're going to raise the temperature back up to 72 degrees C, and that's when Taq polymerase, this enzyme, that's the temperature that it works best at. So it's going to then polymerize or synthesize. It's going to add the bases. So it's going to add all of those extra nucleotides that are hanging out in our little tube and make more copies. Now, every time we do this, we basically are going to double what we have. So we started with one DNA strand, double helix, one DNA double helix. It separates into 2 single-stranded. At the end of this one step, we now have 2 double strands. And when we separate those, we'll have 4 single DNA strands. As we repeat this, we're going to get exponentially more. So you can see here, here's our wanted gene from our template DNA. After our first cycle, we have 4 copies. After our second cycle, we have 8 copies, then 16, then 32. And that's only after the 4th cycle. Each time we are getting more and more and more. So this is like 2 to the 2nd, 2 to the 3rd. This would be 2 to the 4th, 2 to the 5th. You get the gist. So the next one would be 2 to the 6th. If you do this 36 times or 35 times after the 35th cycle, you would get 68 billion copies. And you really do need quite a lot of copies in order to be able to visualize the DNA. So you do, most of the time, you want to do this between 20 and 40 times to make sure you get enough copies. But this is key. This exponential amplification is really, really beneficial. All right, I want to talk about adding this PCR with something called STRs. So STR is a short tandem repeat. This is not part of a gene. And because it's not part of a gene, we can get lots of variation. And I kind of mentioned this a little bit when we talked about mutations. So mutations happen at a kind of regular rate, but if there is a piece of DNA that's not part of a coding gene, if you get a mutation there, it doesn't matter because it's not going to cause the organism to die or have something weird happen to a protein and then that causes them to be more easily eaten or whatever. If it's in this part of the DNA that doesn't copy for anything, it doesn't matter. There's no benefit for fixing it, so there's no selection for it to leave the population. This is how we can tell individuals apart. If you're talking about like a criminal DNA profile, we use 13 locations of these short tandem repeats. Now, a short tandem repeat is anywhere from 2 letters that just repeat over and over to 7 letters that repeat over and over. So usually they will find these short tandem repeats, and then they will make a copy and see how long is the copy, how big is the copy. So I might have 3, for instance, of this AT, and someone else might have, I don't know, 6 or 7. So this person would have twice as many bases as me, and so we would be able to tell that apart looking at the size of the DNA fragments, and that's how we're going to actually tell these apart. So first, we're going to isolate the DNA and amplify. This is PCR, okay? And we're going to use primers that are specific for the locations of those STRs. Then we're going to separate the DNA on a gel. Separation on the gel is going to be based on the size of the fragment. The size of the fragment determines how far it moves in the gel and can let us match people to samples. So say we have, samples that I know that I take from people, and then a sample from a crime scene that I can match it to. And then last, we're going to visualize it in one of a few ways. Now, I want to talk about this part in more detail. The size of the fragment is going to determine how it moves through the gel. When we make a gel, it is literally like a gel. I know you guys have all at least seen Jell-O, I'm hoping. And Jell-O's kind of solid, but kind of not. The gel we use for these is very similar. It's made of something called agar. You can actually make vegan Jell-O that you would eat is made of the same kind of stuff. But in that gel, that agarose gel, you're going to have these little pores, and it depends on how much of the like gel you make, what percent. You can make one that's a very porous gel, one that has lots of holes. And in that case, you use less of your agarose and more water, or you can make one that's got very tiny holes. But what happens is the DNA will start to move through it. And as the DNA moves through it, smaller pieces of DNA can move through it faster. They can kind of weave through those holes quicker. Imagine if you're like walking down the hallway and you've got like, you're very tiny maybe compared to someone who's much bigger. I know when I was in high school, we had this one hallway that was always crowded, and I could get through it a lot faster than some of my friends because I was a relatively smaller person than some of the people that I hung out with. Especially, I had a friend who was on the football team who was a really big guy. It just took him longer to get through because he couldn't like sneak through the gaps between people in the hallway. So that's what these things are doing, is they're going through the gel. The smaller pieces of DNA will get further faster because they can go through those little holes. Now, in this example, we're going to be taking from these, 3 suspects, for instance, and we're looking at this AGAT-STR. So in this location, this person has 7 repeats. Here they have 6, 4, 19, 12, 14. So these are our 2 different STR sites. Remember, I told you DNA profiling, we use 13 STR sites. Once we get the DNA, so we're gonna get our primer, we're gonna copy these things, we're gonna make copies of each of those strands. Then we're going to load it into our gel, and our gel has power source, so we're gonna have an electric current. All DNA is negatively charged, and so it's going to move to the positive electrode. Remember, positive and negatives attract each other. So we're going to load the samples into the wells. We're going to run the electric current. And as we run the electric current, we're going to pull the DNA down the gel into that gel material. The shorter molecules are going to go through faster because they can get through those small little gaps much faster than the larger things. The longer molecules are going to get stopped. Then we're going to need to visualize it. Now, there's a couple different ways to visualize. I'll talk about a few. But this is the basics of what we're going to do. And then what we can do is we can compare the pattern we see in each of these rows with our like crime scene. So say this one would be our crime scene, and then we would have maybe suspect 1 and suspect 2. And we can kind of compare and see which ones look similar. So one way we can visualize is to stain with what's called ethidium bromide. We can view it then with UV light. So the ethidium bromide gets in between the DNA molecules, and then you can see it if you put UV light. If you didn't selectively copy what you're interested in, you might end up with a smear of DNA. So instead of your gel looking like this, where you selectively copied, I have this piece and this piece and this piece were all selectively copied based on the primers. Instead, we might have our gel and our DNA might look like this, like just a smear of DNA. In that case, we can blot it onto a nylon membrane. It's almost like a piece of paper, but it's made out of like nylon or in the like paper fibers. And then we stain it with primers that are specific to the gene of interest. So we can actually stain it with another bit of DNA. This is called southern blotting. There are other techniques where you can actually use antibodies to do something similar. So there are other ways to visualize depending on what you're looking at. But this is just kind of going over those basic techniques so that hopefully you have some background. All right, we are going to end there and do recombinant DNA next time.